Something's been kind of bugging me lately, and I'm betting it's been on your mind too. It's this whole cancer thing, right?
Oh, yeah.
I mean, since the 50s, we've thrown mountains of money and research at it. We see breakthroughs practically every week, but
Right.
the actual death rate from cancer
Right.
hasn't really moved that much. It's like stuck?
Yeah.
And then you think about infectious diseases like 100 years ago.
Huge changes.
We basically doubled our lifespan by figuring out how to deal with those little
It
buggers.
makes you wonder.
It really does. Why is cancer, which seems like, I don't know, pretty basic problem in a way, why is it so hard to crack?
Yeah.
It's almost like trying to explain to someone like a real genius, some astrophysics whiz, why their spaceship won't go to warp speed.
It's that level of head scratching.
It really is. And a bunch of you who we know love to really get into the nitty-gritty of this stuff sent us a really interesting collection of articles and excerpts about a totally different way of thinking about cancer. It's called the Metabolic Theory. And today we're going to dive deep, no messing around, right into the core of this idea.
Yeah.
We want to understand what it's all about and why it might be a total game changer in how we understand this whole cancer puzzle.
It's fascinating how for so long we've been looking at cancer through this very specific lens. Like the dominant theory has been the somatic mutation theory, the SMT.
Right.
So to really appreciate what we're going to unpack today, we need to kind of understand where the established view came from, where it's hitting some walls, and then explore this really cool alternative, the metabolic perspective.
Okay, so let's start with the SMT. Just break it down for us. What's the basic idea?
So the core concept is that cancer starts with mutations, like errors in the DNA inside our cells.
Okay.
And these kind of genetic glitches, if you will, they drive the uncontrolled cell growth and all the crazy things that cancer does.
Okay.
If you think back, it started with scientists like Hans Miltman. He was looking at cancer cells under a microscope, and he saw all these broken chromosomes. He's like, something's clearly not right here.
Yeah.
And then there's Peyton Rouse. He showed that viruses could actually cause cancer in chickens.
Oh, wow.
That was a big deal. It really made people think, hey, maybe something's messed up with the genetic blueprint of cancer cells.
So like a glitch in the operating system.
Yeah, exactly.
And that led to this massive effort, right, to try and pinpoint those specific genetic bad guys in human cancers. Like the whole point of those big projects like the Cancer Genome Project, right?
Absolutely.
Yeah.
The hope was if we could map out the genetic code of all these different cancers, we could find what they call the driver mutations.
Oh, I see.
The specific errors that were causing each type. And then, bam, we could create these super precise targeted therapies like magic bullets
Oh,
that
yeah.
just went after those messed up genes.
Right.
It seemed like a really clear path.
Yeah.
Even James Watson, the DNA guy,
Yeah.
he got frustrated because things weren't moving fast enough. And he was like, hey, we should be looking more at cancer metabolism. He actually said a cancer cell was more like a sick man who needed metabolic help, not some genetically superior Superman that we needed to attack.
That's interesting. So what happened? Why the frustration? Why would someone like Watson suggest looking in a different direction?
Yeah, that's the million-dollar question. So the Cancer Genome Project, it gave us mountains of data, which is amazing, but it didn't quite lead to the cures for everything that people were hoping for.
Oh.
What they actually found was, well, it was just mind-blowingly complex. There were just so many different mutations, and they weren't the same from person to person, even in the same kind of cancer.
Oh, wow.
Even in the same tumor, different parts would have different mutations. It became a nightmare for drug development.
Like trying to find a bad wire in this huge, messy circuit board that keeps rewiring itself?
Exactly. That's a great way to put it.
But didn't they have some success with that leukemia drug?
You're talking about imatinib.
Yeah, imatinib or Gleevec.
Right, Gleevec.
I remember hearing that was like the poster child for how targeting mutations could actually work.
Yes. Imatinib, that was a huge breakthrough. It's incredibly effective for CML, chronic myelogenous leukemia, because it targets a very specific genetic thing, this BCR-ABL fusion protein.
Okay.
But here's the thing, CML is kind of unique. It's what we call a homogeneous cancer,
Okay.
meaning that in most patients, the vast majority of the cancer cells have this same genetic driver.
Oh, I see.
But that's not how most cancers are, especially the solid tumors. They're much more diverse, much more complex genetically.
Yeah.
And a lot of these targeted drugs for solid tumors, they only give you a little bit more time, like a few months,
Huh.
and they're super expensive. And they don't really change the overall outcome for most people.
So the genetic approach, while it's super important, it's shown some promise, it hasn't been the magic bullet we were hoping for.
Right.
Okay, so this is where we rewind back to the 1920s. Enter Otto Warburg and this metabolic theory idea. So tell us, what was his big idea?
So Warburg, he was way ahead of his time. He was looking at how cells make energy, and he proposed that cancer was fundamentally a disease of energy production.
Oh, interesting.
He noticed that cancer cells do this weird thing. They ferment glucose like crazy, even when there's plenty of oxygen around.
Okay.
And usually, cells use oxygen to make energy efficiently in the mitochondria. Right?
Yeah,
So this preference for a less efficient method was a big clue.
it's like a high-performance engine suddenly deciding to run on fumes.
Exactly.
That's a great analogy.
So instead of looking for problems in the DNA, the instruction manual, Warburg thought the problem was with the engine itself.
Yeah, precisely. He thought that this switch to fermentation was like a major defect, and it was because of damaged cellular respiration. He also pointed out that cancer can be triggered by all kinds of things, chemicals, viruses, whatever, and it can pop up in pretty much any tissue.
Right.
So to him, this meant that there had to be a deeper, more fundamental problem, something common to all cells, not just specific to certain tissues.
Okay, and what did other scientists think of Warburg's theory? That seems pretty different from what everyone else was thinking.
At first, people were really interested. But then when DNA was discovered and genetics became the hot new thing, everyone shifted their focus.
Right.
And then, decades later, there was this really influential review by Sidney Winehouse.
Okay.
And for a lot of people, this review basically shut down Warburg's theory. Winehouse argued that there wasn't enough evidence to say that damage to mitochondria was the main cause of cancer.
Okay.
Even Hans Krebs, Warburg's former student who also won a Nobel Prize, thought that while Warburg's findings were important, they were probably a symptom of something deeper, something related to how genes are controlled. And guess what? That's exactly what everyone was focused on at the time. So Warburg's theory kind of faded away.
So genetics ruled for decades. But then the metabolic idea came back, right? What happened? Who brought it back? And with what new evidence?
That's a cool thing about science. Ideas can come back with new evidence. Peter Peterson was a big part of this metabolic comeback. He took another look at Warburg's ideas, but with new technology.
Oh, okay.
And he found some pretty convincing evidence that mitochondria in cancer cells were actually damaged, not just structurally, but also in how they worked.
Oh, interesting.
Peterson thought that maybe these damaged mitochondria were sending out like distress signals to the cell's nucleus,
Okay.
almost like, hey, we're in trouble down here. And then, as a survival mechanism, the cell would start behaving like a cancer cell.
Like an SOS signal from the power plant and the whole factory goes haywire.
Exactly. It's a great way to think about it.
So Peterson and his lab, did they find out how this mitochondrial damage was actually connected to the Warburg effect?
Yes. One of his researchers, Young Hee Ko, made a key discovery. They figured out what was driving the Warburg effect.
Okay.
It was this enzyme called hexokinase 2.
Okay, hexokinase 2.
It's like a gatekeeper for glucose. And in cancer cells, it becomes incredibly efficient at trapping glucose inside the cell.
Like a super vacuum cleaner just sucking up all the glucose.
Exactly.
So this glucose-grabbing enzyme is a major player in this whole metabolic shift. And this is actually connected to the PET scans they use to find cancer, right?
Oh, absolutely. PT scans with FDG, they work because of this increased glucose uptake by cancer cells.
Wow.
The FDG, it's like a modified glucose molecule, and it gets trapped in those metabolically active cells like tumors, and then sends out a signal that the scanner picks up.
So this metabolic difference isn't just some theory in a lab. We're using it every day to diagnose people. So if this metabolic glitch is so common in all these different cancers, what does that mean for treatment?
This is where it gets really exciting. If there's this fundamental vulnerability in the way all these cancers produce energy, we might be able to target that, no matter what type of cancer it is or what specific genetic mutations are going on.
It's like we've been trying to whack-a-mole with all these different genetic mutations, but there might be this one button that shuts down the whole game.
Yeah, that's a good way to put it. Instead of trying to fix all the individual errors in the code, we target the power source.
I like that.
So let's dive into some of the potential therapies that this metabolic understanding is opening up.
Yeah, let's do it. The outline mentions something called 3-bromopyruvate, or 3BP. What's the story there?
So this is where Young Hee Ko's work gets even more interesting. She discovered this molecule, 3BP.
Good.
It's a pretty simple molecule, and it basically blocks hexokinase 2.
Ah, the glucose vacuum cleaner.
Exactly. So by blocking it, 3BP cuts off the cancer cell's fuel supply.
It basically starves it out.
Yeah. Studies in animals have shown that 3BP can shrink tumors, And there's even this amazing case study of a patient, his name was Yivar, who had really bad liver cancer.
Yeah.
And after 3BP treatment, he had this incredible recovery. But getting 3BP into wider use has been a real struggle. Lots of legal and funding roadblocks.
That's so frustrating. It seems like these innovative therapies always face these hurdles. Okay, another metabolic approach we hear a lot about is the ketogenic diet,
Right.
the RKD. We know it's good for lots of things, but how does it specifically help with cancer?
So the idea is that cancer cells are hooked on glucose.
Okay.
They need it to survive, but they're not very good at using other fuel sources, like ketone bodies. The RKD is this super low-carb, high-fat diet that basically forces your body to switch from using glucose to using ketones, which are made from fat.
So it's like going to a restaurant where the only thing on the menu is glucose and suddenly it's not available.
Exactly. That's a perfect way to describe it. And it might not just be about starving the cancer cells.
Oh.
You see, Russell Wilder, he was the first to use the ketogenic diet to treat epilepsy back in the 20s, and he saw how it affected the brain. And it seems that with cancer, the RKD might actually do two good things. One, it makes healthy cells more resilient to stress, which is called differential stress resistance, or DSR.
Okay.
And two, it makes cancer cells more vulnerable to things like chemo and radiation.
So it's like boosting the good guys and weakening the bad guys at the same time.
Yeah, exactly.
The outline mentions some early case studies, like Linda Nebling's work with brain tumors in children and Marianne's story with glioblastoma. What did those studies show?
So these were early studies, so we need to be careful, but they were really interesting. Nebling was working with kids who had really advanced brain tumors where standard treatments hadn't worked.
Oh, wow.
And some of the kids who went on the ketogenic diet actually lived for a surprisingly long time. These were small numbers, but the results were amazing considering how aggressive these tumors usually are.
Yeah.
And then there's Marianne with glioblastoma. It's a really nasty type of brain cancer. And initially, she had a fantastic response to the ketogenic diet, along with standard treatment.
That's great.
Her tumor basically disappeared on scans, but sadly, it came back when she stopped the diet.
Oh, so it shows the potential, but it also highlights that we need to figure out how to make these metabolic therapies work long term.
Exactly.
And what about combining the ketogenic diet with other treatments like chemo, radiation, or even that hyperbaric oxygen therapy?
Yeah, HBOT. Research is starting to suggest that combining the ketogenic diet with other therapies could be really powerful.
Okay.
So by stressing the cancer cells metabolically, and at the same time making healthy cells stronger, the RKD might make radiation and chemo more effective at killing cancer cells.
Okay.
And it could also reduce the side effects of those treatments.
That would be huge.
Right. There's even research by Walter Longo looking at fasting, which has similar effects to ketosis, as a way to protect healthy cells during chemotherapy.
Wow.
And HBOT is really interesting, too, because the high oxygen levels stress the cancer cells that are relying on fermentation. It's all about attacking the cancer from different angles, exploiting its weaknesses.
So like a multi-pronged attack,
Right.
targeting that metabolic Achilles heel. Now, the last thing on our list is repurposed drugs. What's that about?
This is another exciting area. People are looking at existing drugs, drugs that are already approved for other things and are known to be safe.
Oh.
And they're trying to see if they might have anti-cancer effects by affecting metabolism. There are researchers, patient advocates like Jane McClelland and Ben Williams. They've been looking into this.
Oh, wow.
And there's this company, Notable Labs, started by Matthew De Silva. They're doing some really cool work testing combinations of these repurposed drugs on actual patient tumor cells in the lab. They want to find personalized drug cocktails that will be effective for that specific person.
That's incredible.
Right. And because these drugs are already approved, doctors could potentially prescribe them off-label if they show promise in those personalized tests.
Yeah.
It could be a much faster way to get people the treatments they need.
It's like finding hidden treasure in our medicine cabinets. Instead of always looking for something new, we might already have what we need. So we've talked about the genetic theory, its challenges, and this really compelling alternative, the metabolic theory. But why was this metabolic perspective overlooked for so long?
That's a great question. Peyton Ravis, back in the day, he said something really insightful. He said that the somatic mutation theory, with its focus on finding those specific genetic defects, could act like a tranquilizer.
A tranquilizer.
Yeah, like it made people feel comfortable, maybe a little too comfortable. It's as if the complexity of the genetic landscape, while obviously important, maybe distracted us from this more fundamental metabolic issue.
Yeah, it's like the genetic information overload created a smokescreen.
Exactly.
Oh,
The metabolic
see.
target, the way cancer cells make energy, it's actually pretty simple compared to the crazy world of genetic mutations.
Right.
And targeting this weakness could lead to treatments that are less toxic and work for more people. Plus, think about the cost. Things like 3BP and the ketogenic diet are pretty inexpensive compared to those super expensive targeted drugs.
And the stuff you sent us even asks, could all these brilliant minds have been misled by circumstance by focusing so much on genetics?
It really makes you think, doesn't it?
It does.
Maybe the success of imatinib with leukemia, even though it was a very specific case, maybe that made everyone a little too focused on genetics. And they didn't pay enough attention to this more universal metabolic problem.
So to sum up our deep dive today, the metabolic theory of cancer is a really compelling alternative to just focusing on genetics, and it's gaining more and more support.
What's really exciting is that it suggests that we might be able to find more effective, less harmful treatments for a whole bunch of different cancers by targeting this one fundamental weakness in how they make energy.
Absolutely. And we need to remember this isn't about throwing away all the amazing research that's been done on genetics.
Of course not.
It's about recognizing that it might not be the whole story.
Right, and we need more research, especially clinical trials, to really see how well these metabolic therapies work. Things like 3BP and the ketogenic diet.
Totally agree. So, to all of you listening, here's a thought to ponder. We've seen how complex cancer is genetically, and we're still struggling to truly beat it. So, is it time for a big shift? What if we start looking more seriously at the energy side of things, the metabolic foundation of this ancient disease? It's like, what if this seemingly basic flaw in how cells make energy is actually at the heart of this incredibly diverse and devastating illness?
Wow.
It's a pretty profound question, and it really makes you think about the very essence of life itself.
It really does.
We hope you enjoyed this deep dive.
Yeah, thanks for listening.